For centuries, roses have captivated the hearts of people across the world. Adorning gardens and filling the air with their sweet fragrance, they are among the most cherished ornamental plants. Yet, despite their popularity, the true genetic origins and the complex history behind modern roses have remained shrouded in mystery. Until now. A new study has uncovered fascinating details about the genetic makeup of these beloved blooms. By decoding the genetics of modern roses, the research reveals the combined influence of species from both East and West. The findings not only shine a light on the evolution of these flowers, but also offer valuable tools for breeding more resilient and stunning varieties. The story begins in ancient times, when roses were cultivated separately in China and Europe. Each region nurtured its own unique varieties, growing different types of roses that would eventually shape the modern species we know today. But it was the 18th century when a monumental shift occurred. Chinese roses were introduced to Europe, setting the stage for a new era in rose breeding. The introduction of the old blush China rose has long been recognized as a key moment in the transformation of roses. But, as the recent study reveals, there was another crucial player in this evolutionary tale, Rosa odorata. This often overlooked species played a significant role in the fusion of Eastern and Western roses, giving birth to the modern rose varieties we adore today, known for their repeated flowering and diverse forms. At the heart of this research is Samantha, a modern rose variety with striking red petals and continuous blooms. The team of scientists decoded its complex genetic structure, unveiling the intricate beauty hidden within its DNA. Samantha is a tetraploid, explained Professors Hang Jianfei from the Boyce-Thompson Institute, which means it has 4 sets of chromosomes instead of the usual 2, making its genetic structure more challenging to study. Through their groundbreaking work, the research team not only mapped Samantha's genome, but also sequenced the DNA of 233 other rose varieties. From wild species to modern cultivars, they created a genome variation map, a powerful reference for understanding how modern roses evolved and how breeders can continue to refine these beautiful flowers. The implications of this study are vast. Imagine roses that bloom longer, need less care, and are more resistant to pests and diseases. With this genetic knowledge, breeders can develop roses that are not only more beautiful but also hardier, better suited to a variety of climates, and capable of thriving in an ever-changing world. Yet, as the researchers warn, preserving the genetic diversity of roses is critical. Human selection has already shaped the roses we know today, with traits like continuous blooming, and disease resistance carefully cultivated over the years. But with environmental challenges looming, maintaining a broad range of genetic traits will be essential to ensure that roses can continue to flourish in the face of climate change. In a world where science and passion intertwine, the rose is a living tapestry woven from history, biology, and human creativity. Thanks to this new study, the tapestry is now more vivid, more detailed, and more awe-inspiring than ever before.